Well, hello there. Today we're going to talk about a very interesting concept of operons. Our DNA consists of genes. And from the central dogma we know DNA is transcribed to RNA, which is translated to proteins. But in some organisms like bacteria, a few nematodes, the fruit fly, and also viruses, a very interesting phenomenon occurs. Some of their genes can be turned on and off, just like a switch. Such genes are called operons. But before we talk about operon mechanism, we need to understand the structure of the operon. We start off with the promoter, which is recognized by RNA polymers for transcription. The operator is where the repressor binds. And finally, the structural genes are regulated by the operon. But why gene regulation? Well, gene regulation allows organisms to adapt to environmental conditions, somewhat similar to homeostasis. So, what are the types of regulation? Well, regulation is generally of two types. We have positive and negative. Positive control can be positive inducible, where an activator protein, which cannot bind to DNA directly, binds to an inducer, which forms a complex which binds to DNA. The other type is positive repressible. Here, the activator is already bound to DNA, but in presence of a repressor, a complex is formed which prevents the transcription of DNA. Under negative control, also similar types are seen. Under normal conditions, we have the promoter, the operator, and the structural genes, where a repressor protein is bound to the operator, which prevents transcription. In presence of an inducer, the inducer binds to the repressor, rendering it inactive, allowing transcription to take place. Such is seen in lactoperon, where the inducer is allolactose. In negative repressible operons, transcription occurs normally. A regulator gene, however, produces repressor proteins, which do not bind to the operator normally. However, when a co-repressor binds to the repressor, it causes a change in the active site, which prevents transcription. Such is seen in TRP operon, involved in the synthesis of tryptophan. And that basically concludes what an operon is. Thanks for watching.